everybody, it's Mrs. Mancuso. Thanks for joining me for AP Biology Review. I want to talk about how you regulate the expression of genes. So, here's your cell. Inside your cell, you have your nucleus, and inside of here, you got 46 chromosomes. Those 46 chromosomes are diploid. That means you have two sets of 23 chromosomes. One came from mom, one came from dad. Within those 23 pairs of chromosomes, you have all the different genes to express you. But the wild thing is, you only express very few of those genes at any given time. So, for example, in your eye, you're going to turn on rod genes and cone genes and retina genes and vitreous humor genes. And you're going to make all the different parts that make an eye. And so somewhere in these genes are all the directions for doing this. DNA gets transcribed into RNA. RNA gets transcribed into proteins. All of these have protein basis. And so all of those are coded by your DNA, right down to the exact coloring of your eyes, or if you have multiple colors or you have spots or any of those things. That's all determined because of your DNA. But you know what else you have in these 46 chromosomes? You have all the directions to make your fingers and your fingernails and these wrinkles and knuckles and hairs and so many things. But in your eye, they're not expressed. So we have to talk about how is that regulated? How are some things on and some things off? So when you have a section of DNA and it's really on, we call that euchromatin. Euchromatin is DNA that's being expressed. It tends to be loosely wound so that molecules can come in and start transcription. The molecules that initiate transcription are called transcription factors. So in eukaryotic cells, regulation is controlled by transcription factors. And so again, here's your nucleus, here's all these chromosomes, all this DNA all over the place, and a transcription factor says, okay, where do I want to go? Here. And it goes to a very specific gene, and it says, all right, I just want to transcribe this little bit right here. So how does that happen? It's a signal transduction pathway. Here's my whole cell. There's probably a receptor. It could be outside the cell if the ligand is polar. It can be inside the cell if the ligand is nonpolar. But there has to be a receptor. And that ligand signals a signal transduction pathway. So I have reception. I have transduction, which usually involves cyclic AMP and turning on kinases and a phosphorylation cascade. And then the response is that it makes a transcription factor. And the transcription factor goes to your DNA and turns on a gene. So let's say this was this molecule that causes you to make more hemoglobin. Okay, so like let's say your iron level is low and you need more hemoglobin and you're just not carrying enough oxygen around and your body sends this signal. So here's the signal. Here's the transcription factor. It goes right to your hemoglobin gene and it turns on the production of RNA. That RNA is then going to leave the nucleus. Oh, I don't want it to go out of the cell. Sorry. Leave the nucleus, go to a ribosome, and it's going to make hemoglobin. And then that hemoglobin can bind more oxygen and carry more oxygen around your body. So this is how a transcription factor works. It turns on transcription in a specific gene. And when you're doing that, what it's really doing is making your RNA polymerase go to that spot. There's literally thousands of genes here. RNA polymerase will just go where transcription factors are. Now, if something is off, we call it heterochromatin. I like to think of the H as hidden. So this is one way. Coil it really tightly. Transcription factors can't get in there. So if DNA is coiled too tightly, it doesn't work. It also can have a tag on it, and the tag is a methyl. And so if you methylate genes, they're off. So in your eye, you'd have to methylate the genes for making gastric juices because there's no need to make that in your eye. You need to make it in your stomach, but not your eye. So genes will get turned on or turned off or regulated as necessary. Now, in a prokaryotic cell, so these are cells that don't have membrane-bound organelles. In a prokaryote, regulation is carried out by operons. So operons are specific segments of the DNA. So here's one section of DNA. The structural gene is right here. And so when this gets transcribed, it makes RNA, and the RNA goes to the ribosome and makes a protein. So it seems like that's really all I need. But the control is what comes upstream of it. So before it, there's an operator. And before the operator, there's a promoter. And the promoter is where the Tata box is. This is where RNA polymerase joins, RP. It's ready to do its transcription job. And above that is a regulatory gene. The regulatory gene makes RNA and makes a protein. And this protein is called a repressor protein. So here's my repressor protein. This repressor protein actually goes right up here, attaches onto the operator, and blocks transcription. So if I don't do transcription, that means this is stopping right here. RNA polymerase can't get past the repressor protein, so this gene is off. Whatever this is for, you are not making this protein because your RNA polymerase can't get past the operator. So this is one way of controlling genes. Control it by turning it off, by blocking it. This works in what's called an inducible operon. An example of an inducible operon is the Lac Operon. So the Lac Operon makes lactase. Lactase is the enzyme to break up lactose. Lactose is a substrate. Lactose is a disaccharide, and it's a sugar. When this bacteria has lactose sugar, this restriction protein, I'm going to do this pathway in red. This restriction protein comes off and goes, oh, milk is here. Oh my gosh, I love milk. And it comes over here and it binds to this. Now, repressor protein's got nothing in the way. It does transcription, it does translation, it makes lactase. So lactase is only made, this gene is only on when the substrate is present. How awesome is that? No energy is being wasted making lactase when you don't need it. So an inducible operon is normally off, but the presence of a substrate, so here's my substrate, my lactose, turned it on by moving the repressor protein off the operator. Now, how would this stop? Lactase is made. So here's my lactase. It goes over here and it breaks up lactose. When lactose is broken into two molecules, now my repressor protein's like, oh, milk is done, and goes back. That blocks it again and turns it off. So you will run this gene to make as much lactase to break down the milk that you had, and then this will go back when the milk is all broken down. So again, an inducible operon is normally off, but you can turn it on. There are also repressible operons. Repressible, thinking about you can repress it. That means it's normally on, but you could turn it off. So again, I have my structural genes. Those need to be transcribed into RNA and translated into a protein. I have my operator, I have my promoter, and I have my regulatory gene. My regulatory gene makes a repressor protein, but this time my repressor protein is inactive. It doesn't work. So here's RNA polymerase, and nothing's blocking it, so I normally make this protein. And if I make too much of it, you know what this does? Binds to this, makes it active, makes it goes up, block the way. So excess product works as a feedback mechanism to turn off the gene. So normally this was on, making all this stuff. But if I make too much, I activate my repressor protein and I turn it off. All right, thanks for joining me for this review.